Okay, so welcome to this video. In this video, what we're going to talk about is cystic fibrosis and cystic fibrosis medications. Okay, now most of this video is going to be on the pathology of cystic fibrosis. Okay, but we will talk right at the end about cystic fibrosis medications. Okay, now there are a huge number of medications given to people with cystic fibrosis. Now many of them are to treat the symptoms of cystic fibrosis, so for instance antibiotics for the recurrent pulmonary infections. Okay, we're not going to go through all of those drugs, okay, those are for different videos. We're going to look at the specific medications which target the cystic fibrosis transmembrane conductance regulator, which is the protein which goes wrong in cystic fibrosis. Okay, right, so let me outline the structure then for this video. We're going to start off by discussing the symptoms of cystic fibrosis. Okay, then what we're going to talk about is the cystic fibrosis transmembrane conductance regulator, which is the protein which malfunctions in cystic fibrosis. Okay, we'll talk about the different mutations that you can get in the cystic fibrosis transmembrane conductance regulator. Then what we'll do is we'll go through each of the symptoms of cystic fibrosis and we'll look at why having a mutation in the cystic fibrosis transmembrane conductance regulator is going to cause that symptom. Okay, so we'll start with the sweat glands, mainly because that's the simplest one to understand, because quite simply what happens in the sweat glands is cystic fibrosis transmembrane conductance regulator loses its function and that produces the symptom of too salty sweat. Okay, in other portions of the body, it's much more complicated. So for instance, if you take the lungs, the mutation in the cystic fibrosis transmembrane conductance regulator then leads to too thick, too sticky mucus, which then breaks the mucus escalator, okay? And that then leads to the recurrent bacterial infections, which then leads to bronchiectasis and eventual respiratory insufficiency. Okay, so there's much more steps between the initial problem with cystic fibrosis transmembrane conductance regulator and the eventual pathological symptom, basically. So that's why we'll start with the sweat gland, because it really is the simplest. Okay, then we'll go to the respiratory symptoms of cystic fibrosis, then on to the pancreas, which is where the symptoms arise that give cystic fibrosis its name of cystic fibrosis. In fact, cystic fibrosis used to be called cystic fibrosis of the pancreas. Okay, then we'll move on to the intestine, and finally we'll have a little look at the male reproductive tract. Okay, right, so let's start then with a little bit of background knowledge about cystic fibrosis. So, cystic fibrosis is a genetic disease. It's one of the most common genetic diseases among white Caucasians, okay, and it's estimated that approximately 1 in 2,000 people will be born with cystic fibrosis. So 1 in 2,000 live births are affected with cystic fibrosis. Okay, another piece of background knowledge that I'm going to tell you about before we actually move on to the symptoms is that cystic fibrosis also has another name. It's also known as mucoviscidosis, okay, which refers to the fact that cystic fibrosis is going to result in often, all over the place, in many different body places, it's going to result in the formation of very viscous mucus. Okay, and that's going to be the cause of many pathological features of this disease. Okay, so that's why it's also called mucoviscidosis. Okay, right. So, enough background knowledge. Let's now move on to what the actual symptoms then of cystic fibrosis are. Okay, so let's begin with the symptoms. Okay, so we'll start with the symptom that usually actually ends up killing people with cystic fibrosis, which is the respiratory symptoms of the disease. Okay, right. So what happens in the lungs then? Okay, so you get lung disease. Now what happens is that because of the mutation in this cystic fibrosis transmembrane conductance regulator, and I appreciate that I haven't told you anything about that yet, but for now let's just accept that there is this protein called the Cystic Fibrosis Transmembrane Conductance Regulator, which we'll just abbreviate to CFTR, okay, which is responsible for the symptoms of cystic fibrosis. So in cystic fibrosis, what you get is 2 loss-of-function mutations in the CFTR protein. Okay, and we'll see the effects of that later on. Okay, so because of the loss of function of the Cystic Fibrosis Transmembrane Conductance Regulator, what you end up getting is a very thick, sticky mucus produced on the surface of the lungs. Okay, now usually you have mucus on the linings of the lung, which traps bacteria and is then escalated up and out, basically. And this is known as the mucus escalator. Okay, however, this very thick, sticky mucus doesn't get removed from the lungs, basically. It stays put, okay, because the cilia aren't capable of moving it. Okay, now this can lead to the blockage of small airways, so small bronchioles and small bronchi. Okay, in addition, what it leads to is infection. Okay, so bacteria which are usually cleared by this mucus escalator, they're going to remain in the lungs and they're now going to colonize the lungs and they're going to cause respiratory infection. Now, of course, when you get infection of the lungs, what will happen is the immune system doesn't like that at all. It's not good. Okay, so the immune system launches a massive attack against the infection. Okay, so you get further swelling, basically, because you're going to bring in all sorts of inflammatory exudate, and that's further going to help to block up airways. So you're going to get the infection and the inflammation that it causes potentiating this blockage that was already caused by the mucus. Okay, so because you're getting the inflammatory exudate form, that's further going to contribute to the blockage of the bronchi and the bronchioles. Okay, and eventually when you've blocked enough of your airways, that's going to mean that not enough air can actually get into the alveoli anymore, because if an airway is blocked, then no air is going to be able to get through that. Okay, so when you've blocked enough of your airways that you now haven't got enough alveoli actually capable of getting air into them to provide enough oxygen to the body to sustain life, then you die. Okay, and that's known as respiratory failure. So basically, because the respiratory system fails the rest of the body, okay, it's called respiratory failure. Also sometimes called respiratory insufficiency. You might also hear it referred to as that. Okay, right, so that's usually what kills people with cystic fibrosis, the respiratory failure or respiratory insufficiency that's caused by the blockage of the airways. Okay, right, so that's the lung disease. Now what I'm going to mention also is the pancreatic symptoms. Okay, so we'll go to the pancreas next, and the pancreas is where the symptoms that the whole disease is named after arise. Okay, right. So basically, what happens in the pancreas is very similar to what happens in the lungs. Again, you get a very thick, sticky mucus produced in the ducts of the pancreas, and we will discuss all of this a lot more in detail later. We'll see exactly why problems with CFTR leads to this. Okay, again, you get a very thick, sticky mucus produced in the ducts of the pancreas. Okay, now what this then results in is it results in obstruction of the pancreatic ducts. Now, the pancreas is an exocrine gland. It is secreting digestive enzymes into a fluid that then moves into the duodenum. Okay, so let me just draw a little picture of the pancreas so that I've got something to point at. So here is a picture of the pancreas, and it sort of sits within the C portion of the duodenum here. Okay, right. So the pancreas has ducts moving through it, like so, which drain into the duodenum. Okay, so I'll just tell you a little bit about the anatomy now. This is known as the pancreatic duct, so this one here where it's common before we've split into these two separate ones. This is called the pancreatic duct. Okay, this one here is called the main pancreatic duct. Okay, so this is the main division of the pancreatic duct. Once the pancreatic duct splits in two, this one here, which is slightly bigger, is called the main pancreatic duct. Okay, and then the other one's called the accessory pancreatic duct here. So this one is the accessory pancreatic duct. We'll be coming back to this picture later on. Okay, I'll just call it accessory duct. Okay, right. So those are the massive great ducts of the pancreas here, but there are loads of other ducts that will be feeding into them. Okay, so basically, if you are obstructing pancreatic ducts, then what ends up happening is the enzymes which the pancreas is secreting end up blocked. Okay, so if I draw a picture, if we have some duct here, and let's say we've got a great big block here, then all of the enzymes that are being secreted by the portions of the pancreas that drain into the portion of this duct upstream of the blockage, okay, they're all going to remain in the duct here, okay, and that means you're going to get large collections of fluid in the pancreas. Okay, now when you have a large collection of pathological fluid, okay, a pathological collection of fluid, okay, that's known as a cyst. Okay, so in the pancreas, because of the obstructions to the ducts, you get cysts formed. Okay, that is the cystic in cystic fibrosis. Okay, now what's the other part of the pathology of the pancreas? Well, also, parts of the pancreas start to atrophy, basically. So normal pancreatic tissue starts to die, basically. Okay, and it starts to get replaced by scar tissue. Okay, so let's say that this portion here of the pancreas has been replaced with scar tissue, which is what I'm showing in red here. Now, the fancy word for scar tissue is fibrosis. Okay, so portions of the pancreas start to get destroyed and they're replaced with this connective tissue, which is scar tissue, okay, and that's known as fibrosis. So in the pancreas you get these regions of fibrosis, and you also get the formation of these cysts, hence the name of this disease is cystic fibrosis, and it used to in fact actually be called cystic fibrosis of the pancreas. Okay, later on we realised that there are loads of other symptoms that are part of this disease as well, so we dropped the of the pancreas, but we kept the cystic fibrosis, which describes what you find if you look at the pancreas of a person suffering from this disease at autopsy. Okay, right. So, what are the results then of this pancreatic disease? We've described that the actual morphological changes are you get these blockages and they form cysts and fibrotic tissue. Okay, but what's the result of this? Okay, what actual symptoms does this present with? Because the patient isn't going to come and say, I've got blockages in my pancreatic ducts and cysts and fibrotic tissue. Okay, they're going to come saying that they're getting steatorrhea. Okay, so the symptom that this causes is all because the digestive enzymes which the pancreas secretes are no longer actually getting into the duodenum. So remember, the pancreas has a job to perform. Its job is to secrete digestive enzymes into the duodenum, okay, to digest fats and proteins in particular. Okay, so the pancreas secretes enzymes which are involved in digesting fats and proteins in particular. Okay, so when you are now obstructing ducts within your pancreas, then these enzymes are not going to actually be going into the duodenum, so you don't digest fats and proteins properly. Okay, now what does this result in? Well, this firstly results in a condition known as steatorrhea. Okay, now what this means is that you have a lot of fat present within your feces, okay, because if you can't digest the fat, then the fat isn't actually going to be absorbed from the intestine into the blood, okay, so it will remain in the gut lumen and then it will eventually come out in the feces, okay, and the presence of a large amount of fat within your feces is known as steatorrhea. Okay, so generally this presents as very, very large stools, and also they're generally very, very foul smelling. Okay, right. The other big problem of this, of course, is that if you're not digesting fats and proteins and you're therefore not absorbing the nutritional portions of fats and proteins, okay, you're going to get malnutrition, okay, and what this then leads to is failure to thrive, basically. It leads to your growth becoming stunted, Okay, so simply because you're not getting the necessary nutrients to grow properly. Okay, so stunted growth. Right, so those are the big symptoms that result because of the problems with the pancreas. Okay, now there is a nice term for all of this. Okay, so the term that you can use to describe all of this is pancreatic insufficiency. Okay, so basically the pancreas is failing to do its job correctly. Okay, it's very similar to respiratory failure, it's slightly less serious than respiratory failure. Okay, so pancreatic insufficiency means that the pancreas is not sufficiently performing its role. Okay, it's failing the body. It's failing to produce the enzymes that are necessary for appropriate digestion. Okay, well, it's not that it's failing to produce them, it's failing to actually secrete them into the duodenum. Okay, and this causes the problem of malnutrition for the rest of the body. Right, so that's a term that you will often hear used when describing the symptoms of cystic fibrosis, pancreatic insufficiency. Okay, right. The next symptom that I want to talk about is that you can end up getting intestinal obstruction. Okay, so again, the surface of the intestine secretes mucus, okay, and this mucus needs a fluid. Now, if you have cystic fibrosis and you have a double loss of function mutation in the cystic fibrosis transmembrane conductance regulator genes, then you end up with far too viscous mucus being produced on the surface of the intestine. Okay, and this can lead to intestinal obstruction because that mucus is there to lubricate the surfaces of the intestine so that food boluses can easily move through the middle of the lumen. Okay, so if the mucus becomes far too thick and sticky, it fails to perform this role, and instead what you can end up with is obstruction of the intestine with fecal matter and mucus. Okay, so that's another symptom that can result because of cystic fibrosis. Okay, two more symptoms then. Penultimately, male infertility is very, very common in cystic fibrosis. Okay, so what happens is you end up with lack of the vas deferens. Okay, so this is known as congenital bilateral absence of the vas deferens, CBAVD. Okay, so I suppose I should probably write out what that means in full. So the C is for congenital, okay, the B is for bilateral, the A is for absence, the OV doesn't get a letter, and then the VD is for the vas deferens of, oh, I suppose there should have been of the as well, of the vas deferens. Okay, right. So the vas deferens is the tube that leads from the epididymis, okay, and we'll go over the anatomy of this later on, but it leads effectively from the testicles to joining the urethra. Okay, so if you do not have the vas deferens, then basically sperm cannot get from the testicles into the urethra and therefore can't actually be ejaculated. So you produce sperm, but it can't actually be ejaculated. Okay, so that leads to male infertility. Okay, right. However, with the advent of reproductive technology, of course, what we can now do is collect sperm from the testes of people suffering from cystic fibrosis, because the testes don't have the problem, it's the vas deferens that is gone. Okay, the testes are working, so we can collect sperm from the testes, and through artificial methods we can fertilize an egg with those. Okay, right. So this is congenital absence of the vas deferens, CBAVD. Okay, but that's a very, very common symptom of cystic fibrosis, in males anyway. Okay, and then the final thing that I want to discuss is that you end up with very salty sweat, and this is usually the way that cystic fibrosis can be diagnosed, okay? And they've known this for a very long time. People knew that if you kissed your baby on the forehead and that baby tasted salty, that was not a good sign, okay? They knew that would mean that the baby would die pretty quickly. Okay, so in cystic fibrosis, what happens, and we'll see exactly why this happens, you end up with far too much salt, far too much sodium chloride within your sweat, okay? And that is tastably so. So when you taste your sweat, it will taste very salty. Okay, so salty sweat, that's the final symptom of cystic fibrosis that I want to discuss. Okay, right. So that's the initial first section of this over and done with. We've discussed now the basic symptoms of cystic fibrosis. Okay, next up, what we're going to move on to is this protein. The protein that actually goes wrong in cystic fibrosis. Okay, so firstly what I'm going to do is write out in full the name of this protein. CFTR is what we will abbreviate it to from now on, but I do at least once in this video need to write out what this stands for. Okay, so CF is short for cystic fibrosis. Whenever you see CF, that's what it stands for, cystic fibrosis. Okay, and then it's the cystic fibrosis transmembrane. Okay, that's the T. Okay, and then for some reason a letter that then doesn't, a word that doesn't get a letter included in the name is conductance. So it's the cystic fibrosis transmembrane conductance regulator. Okay, and for some reason rather than being CFTCR, it's just CFTR. Okay, right. So that is the protein that's going to get a problem in cystic fibrosis. Okay, now what I want to do is study this protein in a lot of detail, because it is actually a really interesting protein. Okay, it is an ion channel, but you would not think it was an ion channel. It is very different from other ion channels. Okay, really, it's far more like a transporter. It's got the same structure as a huge number of proteins that are transporters, okay, and which usually use ATP to transport things across membranes, which isn't normally what ion channels do, okay. But we're going to see that CFTR is almost just like a broken transporter. It's halfway in between being a transporter and a ion channel, and it really is unique in the way that it functions. So we're going to study that in a lot of detail, if for nothing else, just for interest. Okay, right. But firstly, let's start off just with a little bit of background information. Okay, so firstly, the cystic fibrosis transmembrane conductance regulator. Where is the gene for this? The gene for this is on chromosome number 7. Okay, so one of the autosomes is the important point that you should take from that. Okay, now what does that mean? That means that you have 2 copies of the gene for the cystic fibrosis transmembrane conductance regulator. Okay, so if we let these 2 parallel lines that I've drawn here represent a double-stranded piece of DNA, let's say that this is one of your chromosome 7s, okay, and this is your other chromosome 7. So remember, in all human cells, except of course the sperm cells and the egg cells, okay, you have 2 copies of each of the autosomes. Okay, now the autosomes is a fancy word for chromosomes 1 to number 22. Okay, so we don't include the sex chromosomes in the autosomes, but chromosome 7 is certainly an autosome. Okay, so you have 2 copies of chromosome 7, a maternal copy and a paternal copy. Okay, and both of these chromosome 7s, both your maternal copy, let's say this is the maternal copy, and your paternal copy, let's say this one's the paternal copy, both of them will have genes for the cystic fibrosis transmembrane conductance regulator. Now these genes are known as the cystic fibrosis genes, okay, or the CF genes, okay, and I think I'll colour them in orange here. Okay, now, to get cystic fibrosis, what you need is you need loss of function mutations in both of the cystic fibrosis genes, basically. It's not enough just to get a loss of function mutation in one and then have another one that functions. You need to have lost function in the cystic fibrosis transmembrane conductance regulator in both genes, basically. You need to have very little function of CFTR remaining. Okay, so that means that cystic fibrosis is an autosomal, okay, because it's affecting a gene that is within the autosome, okay. It's autosomal recessive, okay, because you need 2 copies of mutant CFTR genes in order to actually get the disease. You need both copies of your cystic fibrosis genes to have actually been hit in order to get the disease. Okay, right. So, now let's talk about the actual function of the cystic fibrosis transmembrane conductance regulator. Okay, and after we've described the function of it, we can then come back and look at examples of loss of function mutations in the cystic fibrosis transmembrane conductance regulator gene. Okay, but I think it's better to look at the function of it before we actually see the mutations, because otherwise how can we understand what the mutations are actually going to do to its function if we don't know its function? Okay, right. So, let me start off then with its structure, and then we'll talk about what it's actually going to do. Okay, so, firstly, its size is 1,480 amino acids. So it's a reasonably sized protein. Okay, right. So now let me show you its structure. Okay, so it is a protein which sits within cell membranes. Okay, so it's an integral membrane protein. So let's let these 2 parallel lines that I have drawn here now represent the inner leaflet and the outer leaflet of a phospholipid bilayer. So it's not a double-stranded piece of DNA anymore. Instead, it's the inner and outer leaflets of a phospholipid bilayer. Okay, and now I'm going to put the cystic fibrosis transmembrane conductance regulator here. Okay, so it is a single polypeptide, so it's going to have an amino terminus and a carboxylic acid terminus. Okay, and this line that I'm now drawing, this represents the polypeptide. It represents the polymer of amino acids. Okay, so it spans the membrane once. Okay, it starts with its amino terminus in the cytoplasmic side, spans the membrane once. Okay, spans the membrane a second time, a third time, a fourth time, a fifth time, and a sixth time, okay, to make a cluster of 6 membrane-spanning alpha helices. And this cluster of 6 membrane-spanning alpha helices has a special name. It's known as the transmembrane domain 1. Okay, so this is the name for all of these 6 membrane-spanning alpha helices, now highlighted in vivid purple, okay, which are clustered together. Okay, so TMD1 here stands for transmembrane domain 1. Okay, and we'll see the function of transmembrane domain 1 later on. Okay, right. Then, between transmembrane domain 1 and what's going to be transmembrane domain 2, which I haven't drawn yet, you're going to have a number of important domains. So let me put these on. Okay, so here is our first domain here. Okay, and this domain is known as NBD1. Okay, now what does that stand for? That stands for nucleotide binding domain 1. Okay, now this is going to bind nucleotides. Okay, and the specific nucleotide that it's going to bind is ATP, adenosine triphosphate. Okay, now I haven't actually drawn the ATP binding site, but later on when we actually want to look at the function of this thing, I will add on an ATP binding site onto this. Okay, so it has a binding site for a single ATP molecule. Okay, so that's the nucleotide binding domain 1. Okay, after the nucleotide binding domain 1, you have the next domain here, which is known as the regulatory domain, which I'll just abbreviate to R. So R is for the regulatory domain. I should color code this. Okay, so I'll color in the regulatory domain in green here. So the regulatory domain is here in green. Okay, the nucleotide binding domain 1 we'll have here in red. Okay, right. Then after the regulatory domain, what you're going to then have, and again we'll see the function of the regulatory domain later on, okay, after the regulatory domain you're then going to have transmembrane domain number 2, okay, which will have, again, 6 membrane spanning alpha helices. Okay, so this is transmembrane domain TMD number 2. So I think we'll colour in transmembrane domain number 2 in orange here. Okay, and then finally, after transmembrane domain number 2, you'll then have the nucleotide binding domain number 2, which is NBD2, before you then end with the C terminus over there. Okay, so we'll have nucleotide binding domain then in number 2. Okay, so that then is the structure of the cystic fibrosis transmembrane conductance regulator sitting within the plasma membrane of the cell. Okay, right, so this is CFTR. Okay, right, now, its structure is very much so like a lot of other proteins which are transporters, okay? They use ATP to transport things across the membrane. They are active transporters. CFTR is not an active transporter, but it is in the family of active transporters simply because of how much it looks like an active transporter. Okay, so this structure that we've just drawn is typical, absolutely typical, of a family of transporters which are known as ATP, okay, binding, and then cassette transporters. Okay, and for short, ATP binding cassette transporters are often abbreviated down to ABC transporters. Okay, so A for ATP, B for binding, and then C for cassette transporters. Okay, so CFTR, even though it's not a transporter, it doesn't actually function as a transporter. It is in this family of ATP binding cassette transporters simply because its structure is almost identical to all the other members of this family. Okay? But the other members, they actually do function as transporters. They are primary active transporters, excuse me, which bind ATP and hydrolyze it and use the energy released by the hydrolysis of the ATP to pump a molecule across the membrane. Okay. So, cystic fibrosis transmembrane conductance regulator, it actually has another name which refers to the fact that it is within this superfamily of ATP binding cassette transporters. Okay, its other name is ABC for ATP binding cassette transporters, and then it's family C, so there are loads of families of ATP binding cassette transporters. It's in family number C, and then it's actually family member number 7. Okay. So you may well hear CFTR also referred to as ABCC7. Okay, so don't let that confuse you. That's just referring to the fact that it is within this family of ATP binding cassette transporters, okay, even though its function is very different. Okay, so let's now talk about its function. It is in fact a chloride anion channel, okay? So it's going to allow chloride anions to move across the cell membrane, and I might just draw a little circle around there to make it look more like an anion. Okay, so chlorine atoms, once they have gained an electron, basically, okay, it's actually going to be an ion channel that allows chloride anions to move across the cell membrane. Okay, right. So it is the two transmembrane domains, TMD1 and TMD2, which combine together to make an ion channel through which chloride anions can actually move. Now, this anion channel is not open all the time. It has in fact two states. Okay, it can either be in the open state or it can be in the closed state. Now, what causes it to move from being in the closed state to being in the open state? Because it's usually in the closed state, but what causes it to open? Well, it's the ligand molecule. It's a ligand-gated ion channel. Okay, so I'll just write that down. So ligand-gated ion channels, often for short, are abbreviated to LGICs. Okay, L for ligand, G for gated, I for ion, and then C for channel. Okay, so the cystic fibrosis transmembrane conductance regulator is actually an example of a ligand-gated ion channel. Now, what's its ligand? Well, its ligand is ATP. Okay, so what's going to happen is ATP is going to bind to the nucleotide binding domains, and each nucleotide binding domain has a binding site for ATP. Okay, and that's going to cause the ligand-gated ion channel to open. Okay, the cystic fibrosis transmembrane conductance regulator will move to the open state. Now, there's something very different here from other ligand-gated ion channels. Most ligand-gated ion channels bind their ligand, they open, and then when the ligand falls off, they then close back down again. The cystic fibrosis transmembrane conductance regulator is different from that, and it really is unique in the way that it does this. It binds its ligand, opens, okay, when the ATP binds, and then it consumes its ligand. It hydrolyzes the ATP down to ADP and inorganic phosphate. And then once it's done that, it releases those off and then closes. Okay, so this is a ligand-gated ion channel that actually consumes its ligand. How clever is that? When you remember all of that crisis in neurotransmission where you've got neurotransmitters being released and binding to ligand-gated ion channels on the postsynaptic membrane, which then open. Okay, and then you have to have some other means to remove the neurotransmitter. For instance, acetylcholine is broken down by acetylcholinestrases. Wouldn't it have been simpler if the ligand-gated ion channel for acetylcholine was actually capable of breaking down its ligand itself? It wasn't, okay, but these things are actually capable of breaking down the ligand that they use themselves. Okay, and really, they are an example of a broken transporter. They've still got some of the properties of the transporter, for instance, the fact that they hydrolyze the ATP, okay, but they're not pumping things up the electrochemical gradient anymore. They're just opening a channel which then allows things to move in any direction down the electrochemical gradient. So everything's passive, whereas before, if it was actually functional as a transporter, it would have been against the electrochemical gradient. It would have been very much so active transport. Okay, right. So, let's now talk about how you actually then open the cystic fibrosis transmembrane conductance regulator. So I want to discuss what actually causes this thing to open, because at the moment what I've drawn you is definitely closed. Okay, so I want to talk to you about how this thing actually opens. Okay, right. So the first thing that has to happen, long before ATP comes along and binds, is this regulatory domain has to be phosphorylated. If the regulatory domain is not phosphorylated, then the thing is just going to sit in the closed state, you know, forever. Okay? It will not start functioning at all until this regulatory domain gets phosphorylated. Now, the regulatory domain is going to be phosphorylated by protein kinase A. pKa. Okay, so this is protein kinase A. Okay, so all sorts of receptors can activate protein kinase A within a cell. Okay, there are loads of receptors which then lead to the GS cascade, which then leads to the activation of adenylyl cyclases. Cyclic AMP goes up, activates protein kinase A. Okay, then what will happen is the protein kinase A is going to phosphorylate that regulatory domain. Okay, so I'll draw this here. So this regulatory domain is going to end up with a phosphate group added onto it, like so. Okay, now, I just want to add something else in here. I need to get something off my chest. Okay, protein kinase C. Protein kinase C is not capable of phosphorylating the regulatory domain in the same place as protein kinase A phosphorylates it. Okay, protein kinase A can phosphorylate the regulatory domain of CFTR, and then after you've phosphorylated that regulatory domain, this thing is now ready to function. It's ready to bind ATP and then open if ATP comes along. Okay, protein kinase C can phosphorylate the regulatory domain, but it's not capable of actually phosphorylating it in the right place to turn this thing on. Okay, so what I mean by that is if you don't have protein kinase A active, but you do have protein kinase C active, protein kinase C will phosphorylate the regulatory domain at other sites. Okay, but these phosphorylations do not turn this thing on. Okay, so if protein kinase A isn't active, even if you've got protein kinase C active, this thing will stay in the closed state always. It won't start going into the state where ATP can open it. Okay, so you need protein kinase A active in order to get the phosphorylation of the regulatory domain in the correct position for this thing to now turn on and be ready for ATP to open it. Okay? So, protein kinase C's regulatory role on this is more about potentiating protein kinase A's activation of it. Okay, so that's a little bit more complicated, but we know that protein kinase C alone is not enough to activate the CFTR protein. Okay, so I've just got that off my chest now. So protein kinase A is needed to phosphorylate this regulatory domain here. Okay, once you've phosphorylated that regulatory domain, we are now ready. We are now ready to start binding ATP. So before that was phosphorylated, the nucleotide binding domains would not bind ATP. Now they're ready to start, okay? So, the first thing that's going to happen is nucleotide binding domain number 1 is going to bind ATP first. Okay, so I'm going to draw another picture down here. Now, is this visible in the camera? Okay, let me just move it up a little bit. Okay, right. So, we'll have the cell membrane here, like so. Okay, then I'll draw the transmembrane domain 1 again here. Okay, here's the amino terminus. Okay, now we've got nucleotide binding domain number 1. Now I said I was now going to draw it with an ATP binding site. That's what that little invagination that I put there is supposed to represent. Okay, so previously I forgot to put that on, but that's now representing our ATP binding site. Okay, so I'll keep to the same color coding. Here is nucleotide binding domain number 1 in red. Here are the 6 membrane spanning alpha helices of transmembrane domain 1. Okay, here is the regulatory domain here, which now has a phosphate group attached to it. Okay, so I'll colour that in. Okay, so that's essential. That phosphate group is now going to remain on there throughout this entire opening cycle. Okay, it's necessary before we can even begin this opening cycle. Okay, then we'll have transmembrane domain number 2 here. Okay, and then I'll stop there before I draw nucleotide binding domain number 2, and I'll firstly discuss the ATP binding to nucleotide binding domain number 1. Okay, so it's quite simple. An ATP molecule, which is going to be simply shown as a sort of egg shape like that, which will colour in a special colour. What will show up? Blue, might know, blue's our nucleotide binding domain. I don't want to do it in vivid purple, I'll do it in turquoise, we haven't used turquoise. Okay, so here's an ATP molecule bound to nucleotide binding domain number 1. So this here, this is adenosine triphosphate, ATP. Okay, right. Now, something else is going to happen once ATP binds to nucleotide binding domain 1. Okay, which is that the nucleotide binding domain number 2, which I've just realised you can't see, this blue domain up here, this is now going to bind to the nucleotide binding domain number 1 once it has got the ATP bound. So they're going to dimerize, basically. So I'll draw this here. Whoops, and I've forgotten something else that I wanted to show. Let me just try and repair that. Okay, so I'm also showing that nucleotide binding domain number 2 has an ATP binding site here. Now there's no ATP bound to that yet. Okay, so here is nucleotide binding domain number 2. Still, of course, connected up to transmembrane domain number 2 there, and here's the C terminus. Okay, so that was supposed to be a little invagination there, which is the ATP binding site of nucleotide binding domain number 2. But there's no ATP bound there yet. Okay, so let me summarise what has just happened. We have phosphorylated the regulatory domain of the cystic fibrosis transmembrane conductance regulator. That turns the whole thing on. That makes it so that the nucleotide binding domain 1 can now bind ATP. Okay, the nucleotide binding domain number 1 has now bound ATP, and the nucleotide binding domain number 2 has now dimerized with that. Okay, now the thing is not open yet, so the transmembrane domains here, TMD1 over here, okay, and TMD2 over here, these are not yet in states where they can open, okay? They're not yet in a state where they are open, okay? So there's not an open chloride channel yet, okay? We're going to have to do more than that before we're in the open state. Okay, so what now is going to happen, the next stage, is that another ATP molecule is going to come and bind to the other nucleotide binding domain. Okay, so now that these two have dimerized, another ATP molecule can come and bind to nucleotide binding domain number 2. Okay, and once an ATP molecule has bound to nucleotide binding domain number 2, now the thing opens. Okay, so once you've got 2 ATP molecules bound there, now the thing is going to open. Okay, so this now will go into the open state. So the transmembrane domains will change conformation enough that the thing goes into the open state. Okay, right. So it will remain in that open state for a bit, and then what's going to happen? Well, it's going to hydrolyze one of these ATP molecules. Now, it's important to understand that it's only going to hydrolyze one of them. It's not going to hydrolyze both of them. It's going to hydrolyze one of them. And the one that's going to get hydrolyzed is the one that bound most recently. The one that bound to nucleotide binding domain number 2. Okay, so this one's going to get hydrolyzed. Okay, so it will be broken down to ADP and an inorganic phosphate, and that inorganic phosphate is going to be released. Okay, so let me show this. Okay, so once again we'll draw the picture out again. So here is transmembrane domain 1, and I'll just move this up a little bit. Okay, so this is transmembrane domain 1, consisting of these 6 membrane spanning alpha helices. Then we've got nucleotide binding domain number 1 here, with its ATP binding site still with ATP bound, so it has not hydrolyzed its ATP molecule. Okay, and we'll have this in red again, like so, and then I'll show transmembrane domain number 1 again in vivid purple here. Okay, and we'll have that ATP molecule shown in turquoise here. Okay, then after nucleotide binding domain number 1, we have the regulatory domain here with its phosphate group attached. Okay, we'll colour in the phosphate group again in vivid purple, and the regulatory domain in green here. Okay, then after the regulatory domain, we have transmembrane domain number 2 here. Okay, so this is TMD number 2, and we'll colour that in orange. Okay, right. Then after transmembrane domain number 2, we know we've got the nucleotide binding domain number 2 down here, and this is where things have changed. Okay, so nucleotide binding domain number 2 has just hydrolyzed its ATP molecule that was bound to it. Okay, so it now has an ADP molecule bound there instead. So I will colour in ADP in what colour shall I have? I'll do it in yellow maybe, we haven't used yellow. Okay, so in yellow here, this is no longer an ATP molecule, this is an ADP molecule. Okay, so that is adenosine diphosphate. It's had one of its phosphate groups cleaved off. And of course, the phosphate that has been cleaved off has gone off into the cytoplasm. Okay, right. Now, that does not cause it to close, not yet, okay? It's still in the open state. So both of these states, both where you've got 2 ATP molecules bound here, within the nucleotide binding domain dimer like this, okay, and the state where you've got 1 ATP and 1 ADP bound, those are both open state chloride channels, which are allowing chloride anions to move across the cell membrane, okay? And what is now going to happen is this ADP molecule is going to cleave away from nucleotide binding domain number 2, and then, that's the snap, then it closes, okay, and no more chloride anions can move across the cell membrane. Okay, and then where are we back to? We're back to that state where we've got nucleotide binding domain number 1 with an ATP molecule bound to it, and it's dimerized with the nucleotide binding domain number 2, which has nothing in its nucleotide binding site here. Okay, so what can now happen is either the ATP molecule can dissociate from the nucleotide binding domain number 1, and then you go back to this state up here, okay, or another ATP molecule can come and bind in here, okay, and then you go back to this state, okay, you go back to the open state, and then you go round another cycle. So basically it goes round and round in this cycle of opening and closing and opening and closing, and all the time consuming a molecule of ATP. So every time it opens, it consumes a molecule of ATP and hydrolyzes it down to ADP and then organic phosphate. So it's a really weird ligand-gated ion channel. ATP binds, gets hydrolyzed, and whilst the ATP is bound and being hydrolyzed, the channel is open, and then once the ATP has been hydrolyzed and the ADP and the inorganic phosphate have cleaved off, then it closes again, and then it's ready to go round in the cycle again. Okay, so it is a ligand-gated ion channel that consumes its ligand, it consumes the ATP, which is on the cytoplasmic side. That's another important point, that the ligand needs to be on the cytoplasmic side. Okay, remember there's a very low concentration of ATP in the extracellular fluid. It's not a good sign if you've got ATP in your extracellular fluid. It means that cells have been dying and releasing ATP into the extracellular fluid. Okay, right. So the final thing that I just want to say is that how often this thing is going to cycle round and go into the open state is going to depend on the cytoplasmic concentration of ATP. If you've got loads of ATP, you'll be going around this cycle all the time, and you'll spend a lot of time in the open state. And if you've got very little ATP, you're not going to be in this state very often, and you're going to be in the closed state most often. Okay? And remember also that even to start the whole cycle off, you needed that phosphorylation on the regulatory domain of the cystic fibrosis transmembrane conductance regulator by protein kinase A. Okay, right. So I think we will call it there for this video. We've discussed a lot. In the next video, what we'll do is we'll continue on with the cystic fibrosis transmembrane conductance regulator. We'll talk about the different mutations that can occur in the cystic fibrosis transmembrane conductance regulator, which all cause loss of function of the cystic fibrosis transmembrane conductance regulator. Okay, and then what we'll move on to is how having a mutation in this actually leads to the symptoms, basically. And we'll start off with why does loss of function of the cystic fibrosis transmembrane conductance regulator cause too salty sweat, okay? And then we'll move on to the lung.